Hello viewers, today we will study about T cells and I have planned this lecture into two parts. So we begin with part 1. In part 1, we will talk about only development and differentiation of T cells. So how the story of T cells start? It begins with hematopoietic stem cells which travel from fetal liver to bone marrow. And here in bone marrow, they differentiate into multipotent progenitors, and only a small portion or subset of this multipotent progenitors migrate towards thymus and differentiate into early thymic progenitors. So those cells which migrate and differentiate into early thymic progenitors. Here we should remember that thymus does not contain any cells which it can make itself and convert into T cells. All those cells which are traveling from hematopoietic stem cells to bone marrow and bone marrow to thymus, only these cells will be educated and trained by thymus. So thymus itself do not make any cells. Now, these early thymic progenitors have the capacity, they are multipotent, so they can generate into T cells, B cells, natural killer cells, myeloid cells, and dendritic cells. So ETP represents a small and heterogeneous subset, and that have the ability to proliferate massively. ETP, when attracted by chemokines. It's an interesting story. These early thymic progenitors will be attracted by chemokines, that is CCL19 and CCL21. When they will be attracted by these, they enter the thymus via the corticomedular junction. So through corticomedular junction of thymus, these ETP enters. When this ETP enters the thymus and they encounter large number of ligands for notch receptors. What are notch receptors? They are short signals. These short signals in presence of ligands welcome these ETPs, and there are certain growth factors and interleukin 7 in presence of these ETP trigger these things trigger and support the differentiation of and proliferation of ETP. Here ETP start differentiating and proliferating, and this stimulates the development of this starts the development of T cells. So ETPs when enter the thymus and encounters these ligands for the notch receptors as well as growth factors, interleukin. This gives rise to differentiation of ETPs into T cells. Differentiation of T cells takes place in 6 steps. Let's study each step. First is double negative 1. In double negative 1, the thymocytes, what happened to this ETP and how these thymocytes starts differentiating. This happens in cortex, and this double negative do not express CD4 or CD8. They are double negative because they are not expressing cluster of differentiation, that is 4 or 8. These are very important. Cluster of differentiation is very important for T cell development. These are the sites which are very essential for the functioning of T cells. Now, when this double negative converts into double negative 1 converts into double negative 2, these steps are there for us to better understanding of development and differentiation of thymocytes, that is T cells. DNA1 acquires, that is double negative 1 acquires CD25 and CD44, that is cluster of differentiation 25 and cluster of differentiation 44. These are gateways, and when these are acquired, then double negative 1 gets converted into double negative 2. We start calling double negative 1 to double negative 2, and now these cells start expressing proteins. What proteins they are expressing? They are expressing proteins for TCRs. What are TCRs? TCRs are T cell receptors. These are the receptors which are going to encounter which are going to take up the antigens, receive the antigens. So what happens? These T cell receptor gene arrangements such as recombinant activating genes, that is RAG1 and RAG2, gets activated here, while some proteins are also expressed which are necessary for the assembly of TCR. What is TCR basically? It is a chemical combination that a cell will be expressing, and it will act as a receptor to receive the antigen, to combat the antigen. Where we said that lock and key kind of arrangement happens, that is, antigen will come and lock into this TCRs. So these TCRs, how they are going to develop, how these T cells will differentiate and acquire these T cell receptors, and this start happening in case of double negative cells, and assembly will start, that is TCR assembly will start by signaling CD3 chains, kinase, certain kinds of kinases, phosphatase, such as LCK, that is lymphocyte specific protein kinase, ZEP7, that is zeta chain associated protein, LAT linker. LAT is linker for activation of T cells. These play a very important role, and while we will be discussing certain kind of diseases, we will see that these molecules are very important. At present, since we are not studying diseases, we are just remember these molecules play a role in differentiation of T cells. Now, next thing is double negative 2 cells will differentiate into third step, that is double negative 3. What happens in this case, why we are calling it double negative 3? In this case, the cells which have undergone double, which were not the thymocytes, which we were calling double negative 2, will fall, can take up two routes. One route is either they can express the alpha or beta chains, that is, I told you that assembly of T cell receptors have already started in case of DN2. So in DN3, what happens, this assembly can assemble and take a alpha or beta chain arrangement and follow the process of selection to CD4 or CD8. That means alpha beta chains will be assembled in such a way that either they will acquire CD4 cluster of differentiation, or they can acquire CD8 cluster of differentiation. If the double positive 2 can follow two routes. One, they acquire these things, CD4 or CD8, or other, they express gamma or delta chains and take a different route and follow, become some kind of special lymphocytes. So coming back to DN3, if they follow the route of alpha beta chain and expression of beta chain in case of TCR, that is T cell receptor, it leads to this double positive state. And in the case of double positive state, now these stages are the development of thymocytes, T cells. So what happens when alpha beta chain is acquired by thymocytes, and they are the T cell, and these alpha beta chains are for T cell development of T cell receptors. So expression of CD4 or CD8 molecules may take place, and thus the cells convert into double positive, because now they are developing cluster of differentiations 8 or 4. Now, this double positive constitutes the largest population of cells in the thymus. Why DP are largest population of cells, we will know later. At double positive state, cells enter a control point. Why they are more in population? Because lot of soldiers are prepared. Lot of soldiers are in the process of preparing to fight. That means at DP stage, a selection procedure, first selection procedure has to take place. Now they have to put for the test whether they are ready to fight. So how this selection procedure takes place? With this selection happens with functional T cell receptors. Now, functional T cells receptors have to give a test. What test they are going to give? They have to be tested against self-peptides. Self-peptides which are at the intermediate affinity and avidity. That means the close ones are closer peptides are given to test. That means first test is given to double positive thymocytes. First test. and in this first test, they are now, this double positive cells will have the epithelial cells of thymic cortex, and to test by presenting these epithelial cells will present their own peptides in the context of class 1 or class 2 molecules. That means class 1 HLA. What is class 1 HLA? HLA is human leukocyte, and that HLA, I have put this human leukocyte antigen, and this HLA and HLA1 and HLA2. I have put a whole video on these molecules. Please first see it. If you see that video, then this point will be more clear to you. It's that how human leukocyte antigen class 1, class 2 molecules will test these. Only a fraction of double positive cells that express T cell receptors with intermediate affinity for these antigens persist for survival signal. That means at this first stage of test, that is why double positive are major in population, because they will be tested and most of them will be removed. So DP cells, double positive cells, incapable of binding to HLA1 or HLA2 will undergo apoptosis. At the first test, they were failed. Those who failed will undergo apoptosis. Then the second test will be that is single positive. What happens in single positive selection? It allows the differentiation of double positive thymocytes towards a single positive population that is restricted to HLA1 or HLA2. When they were given first test, when double positive tests were given, they were tested for HLA, HLA as a one, that means HLA1, HLA2. If they are binding to both, they were apoptosis was not there. Now, second, if single positive, what happens? Those who will bind to HLA1 and those who are binding to HLA2 will persist, and others all will be removed. Subsequently, single positive cells enter the medulla of the thymus. When they are selected, they are tested again. Then all those which were showing single positive, that means either they are attaching to class 1 or class 2, and others were all removed. That was the second control point. And what happens when after this single positive selection, negative selection takes place, and at the medulla, positively selected thymocytes are exposed. First, they were exposed to immediate peptides, which were close to they were tested for epithelial thymic cells. Peptides. Now, what happens? Diverse kind of peptides, self peptides, are given to them. That is, second selection is going to take place, and diverse set of antigens will be these thymic cells will be tested for diverse set of antigens. So single positive cells, that is single positive thymocytes with high affinity or avidity for binding self peptides presented on HLA1 or HLA2 are eliminated by apoptosis. That means those which are binding for self peptides will be eliminated. They will be cleared. Now, why this is negative selection? Because when you are binding for self peptide, that means TCR receptors which are binding with self peptides, that will be removed. And because this is to avoid the self-destruction, autoreactive cells, this is to avoid self-destruction. So TCRs which are binding to self-peptides, that means they are not able to recognize self, they are binding with their own self. That means they can remove, they can react with their own self, and they can kill their own self. So at this point, they are not properly educated. So negative selection happens, that you are binding to your own self and treating it as enemy. So these T cell receptors which are showing this tendency will again be cleared. Apoptosis will happen. So cells that survive negative selection, that means those who have not bound with the self polypeptides will get and convert into, will differentiate and convert into naive T cells. And these naive T cells are the cells which are selected for fighting, which will be sent to borders. But when they are they are called naive T cells because still they have not encountered any enemy. They have not come in contact with enemy, and that is they have not come in contact with any non-self antigen. So they are having TCRs, but these TCRs has still not done any work. So these naive T cells will now, when they are naive only, then they will be sent to borders. That means they will leave the thymus and migrate continuously to the secondary lymphoid organs to be primed and differentiate into effector cells. There, if they encounter to antigens, they will be primed and then they will start proliferating. How they will be primed and how the proliferation start, that we will do in next lecture. Here we talked about how these 6 steps happens and how these cells are selected and differentiated and trained to become, these cells are educated and trained to become and differentiate into T cells. And what are the different functions of T cell? We will be talking it about in the next lecture. Thank you so much.